Aseptic processing is a critical method used in the pharmaceutical industry. It's designed to manufacture sterile drug products without a final, terminal sterilization step. Instead of sterilizing the product in its final container, this process relies on a different principle. It brings together previously sterilized components, the drug solution, the container, and the closure, in an exquisitely controlled environment. This environment is specifically engineered to be free from microorganisms. The primary goal is simple yet profound: to prevent microbial contamination from ever reaching the product. This makes aseptic processing the unseen guardian that protects patients from harmful infections when they receive injectable medicines, ophthalmic solutions, or other sterile therapies. The importance of this process cannot be overstated. Many modern medicines, particularly complex biological products like vaccines, monoclonal antibodies, and cell therapies, are sensitive to heat. They just can't withstand the high temperatures or radiation used in traditional terminal sterilization methods, as these harsh conditions would destroy their delicate molecular structures and render them ineffective or even harmful. Aseptic processing provides the only viable pathway for bringing these life-saving and life-altering treatments to patients safely. It's the foundational technique that underpins much of the innovation in biotechnology, ensuring that the most advanced therapies are not only potent, but also pure and safe for administration directly into the human body. Think of it as a highly choreographed performance where every element plays a vital role. The air must be pure, the surfaces must be sterile, the equipment must be flawlessly prepared, and the personnel must move with deliberate, trained precision. A single misstep, a tiny breach in the sterile barrier, can lead to microbial contamination of an entire batch of product. The consequences of such a failure are severe, ranging from costly product recalls to, most tragically, serious patient harm or death. Therefore, the successful execution of aseptic processing is a fundamental responsibility of any pharmaceutical manufacturer, a non-negotiable commitment to patient safety and product quality that is enforced by regulatory bodies worldwide. This essay will delve into the core principles and practices of modern aseptic processing, with a specific focus on the guidelines set forth by the United States Food and Drug Administration. We'll explore the meticulous design of clean rooms, the stringent requirements for personnel and equipment, and the step-by-step execution of the process itself. Furthermore, we'll examine the primary sources of contamination and the robust strategies employed to mitigate these risks. The journey through this controlled world reveals a discipline that is part science, part engineering, and part art, all dedicated to the singular mission of delivering sterile medicines safely to the patients who depend on them. The US FDA's Guidance for Industry Sterile Drug Products Produced by Aseptic Processing, Current Good Manufacturing Practice, or CGMP, serves as the foundational document for manufacturers. This guidance isn't just a simple checklist, but a comprehensive framework built on established scientific principles. Its primary objective is to ensure that drug products intended to be sterile are produced under conditions that robustly prevent microbial contamination. The guidance emphasizes a proactive, risk-based approach, urging manufacturers to understand their processes intimately, identify potential hazards, and implement effective controls. It establishes the expectation that sterility is not something to be assumed, but must be actively designed into the facility, the equipment, and the operational procedures from the very beginning. A central tenet of the US FDA guidelines is the concept of control. Control must be demonstrated and maintained over every aspect of the manufacturing environment. This includes the air quality, the differential pressures between rooms, the temperature, and the humidity. It even extends to the physical state of the facility, demanding that surfaces be smooth, non-porous, and easily cleanable to prevent the accumulation of contaminants. The guidelines stress the importance of a well-defined and validated system for monitoring these environmental conditions continuously. Any deviation from the established parameters must trigger an immediate investigation and corrective action, ensuring the sterile core of the operation is never compromised. The US FDA also places immense focus on the separation of operations to prevent cross-contamination. The design of a facility must logically flow from less clean areas to the cleanest areas, with physical barriers and procedural controls at each transition point. For example, personnel and materials should follow distinct one-way paths into the aseptic processing core. The guidance underscores the need for robust airlock systems for both personnel and equipment, which act as buffers between zones of different cleanliness levels. This meticulous segregation is critical to protecting the Grade A or ISO 5 zone, where the sterile product, containers and closures are directly exposed to the environment from contamination originating in surrounding areas. Ultimately, the FDA's philosophy is one of total control and verifiable assurance. It's not enough to simply perform the steps of aseptic processing. A manufacturer must be able to prove that the process was in a state of control throughout the entire batch production. This is achieved through rigorous validation, qualification, extensive environmental and personnel monitoring, and detailed batch records. The guidance requires a culture of quality where every operator understands their role in maintaining sterility. It pushes the industry towards adopting advanced technologies like isolators and restricted access barrier systems, or RABs, which physically separate the operator from the critical zone, further minimizing the primary source of contamination: people. The heart of any aseptic processing facility is the clean room, a specially constructed environment where airborne and surface contamination is kept to an absolute minimum. The US FDA, aligning with international standards like ISO 14644-1, mandates a classification system for clean rooms based on the maximum allowable concentration of airborne particles. The most critical area, where sterile materials are exposed, is designated as Grade A or ISO 5. This zone must maintain exceptionally clean air, with strict limits on both viable, meaning microbial, and non-viable particles. This is typically achieved using a unidirectional flow of high-efficiency particulate air, or HEPA, filtered air, which constantly sweeps particles away from the critical processing zone, creating a super clean environment. Surrounding the Grade A zone are support areas with progressively less stringent cleanliness classifications. The area immediately adjacent to the Grade A zone is the Grade B, or ISO 7 clean room. This room serves as a background environment for the aseptic core and is where operators, fully gowned, perform final preparations before entering the critical zone. Beyond this lies the Grade C, or ISO 8 area, often used for solution preparation or equipment cleaning. Finally, Grade D, or unclassified areas, serve as the outermost layers, used for initial gowning and material entry. This cascading classification system is designed to create a buffer, ensuring that contamination from the outside world is systematically filtered out as one moves closer to the product. Maintaining these classifications requires a sophisticated heating, ventilation, and air conditioning, or HVAC system. This system is the lungs of the facility, supplying a massive volume of HEPA-filtered air to the clean rooms. A key design feature is the creation of positive pressure differentials between adjacent rooms. The Grade A zone is kept at the highest pressure, so air always flows outwards from the cleanest area to the less clean areas, never the other way around. This prevents contaminated air from adjacent spaces from infiltrating the critical zone. These pressure differentials are continuously monitored by instruments and are a critical parameter that must remain within specification during all manufacturing operations. Beyond air quality, the physical design of the clean room is paramount. The US FDA guidelines require that all surfaces, walls, floors, and ceilings be smooth, impervious, and non-shedding. There should be no cracks, crevices, or ledges where contaminants can accumulate. Corners are coved to facilitate effective cleaning and disinfection. All equipment and furniture within the clean room must be made from materials like stainless steel that can withstand repeated sterilization and cleaning agents without degrading. The entire design philosophy is centered on creating a sanctum that is not only clean but can be reliably and repeatedly cleaned and sanitized to a microbial-free state before each production run. Humans are the single greatest source of microbial contamination in a clean room environment. An average person sheds millions of skin particles and microorganisms every minute, making personnel control the most critical challenge in aseptic processing. The US FDA guidelines are therefore exceptionally strict regarding personnel practices. Before entering the aseptic core, operators must undergo a meticulous and validated gowning procedure. This process is designed to completely encase the operator in a sterile, non-shedding barrier. The gowning typically occurs in a series of dedicated rooms, moving from less clean to cleaner areas, with specific garments added at each stage to minimize the transfer of contaminants. The gowning process itself is a precise ritual. It begins with the removal of all street clothes and personal items, followed by donning facility-specific scrubs and shoes. In the next airlock, operators will don a hood, a mask, and initial boot covers. The final gowning step, often occurring just before entry into the Grade B area, involves putting on a sterile one-piece coverall, sterile goggles, a second pair of sterile boot covers, and sterile gloves. Every item of this outer garment is sterile and must be handled using an aseptic technique to avoid contaminating its outer surface. The process is so critical that operators must be formally qualified, demonstrating they can gown without contaminating their gloves or the exterior of the suit. Once inside the aseptic processing area, operator conduct is rigidly controlled. Movements must be slow, deliberate, and minimized to reduce the generation and dispersal of airborne particles. Rapid motions create turbulence in the unidirectional airflow, potentially drawing contaminants into the critical Grade A zone. Operators are trained to never position their bodies or any non-sterile items between the HEPA-filtered air source and the exposed sterile product or components. They must maintain an awareness of the first air, the clean air coming directly from the HEPA filter, and ensure it is never obstructed from bathing the critical sites in a protective curtain of sterile air. Furthermore, practices that are normal elsewhere are strictly forbidden. Talking should be minimized, as it generates aerosols. Touching any part of the face or adjusting goggles is prohibited. If an operator's sterile glove touches a non-sterile surface, it must be immediately changed or sanitized using a validated procedure. The US FDA expects companies to have a robust personnel monitoring program, which includes routinely taking surface samples from operators' gowns, gloves, and face masks after they complete their work in the aseptic area. These samples are then incubated to check for microbial growth, providing objective data on the effectiveness of gowning procedures and operator discipline. Just as the environment and personnel must be controlled, all equipment and components that enter the aseptic processing area must be rendered sterile. The US FDA guidelines mandate that any surface that will directly or indirectly contact the sterile drug product must undergo a validated sterilization process. The most common and reliable method for sterilizing equipment, such as stainless steel holding tanks, piping systems, and filling needles, is steam sterilization. This is typically accomplished using a large specialized autoclave or through a steam-in-place or SIP process, where saturated steam under pressure is circulated through the equipment assembly to kill all microorganisms. The validation of these sterilization cycles is a rigorous data-driven exercise. It involves placing biological indicators, which are standardized preparations of highly resistant bacterial spores, like Geobacillus stereothermophilus, in the hardest-to-reach locations within the equipment. After the cycle, these indicators are tested for growth. A successful validation study must demonstrate a complete kill of these resilient spores, providing a high degree of assurance that the process is effective. Temperature mapping studies are also conducted to ensure that every part of the equipment reaches the target sterilization temperature for the required duration. These validation activities must be documented thoroughly and re-evaluated periodically. For components that cannot withstand the high heat of steam sterilization, such as rubber stoppers or some plastic containers, alternative methods must be used. These might include dry heat sterilization in specialized ovens, which uses higher temperatures for longer periods, or sterilization with gases like ethylene oxide or vaporized hydrogen peroxide. Each of these alternative methods must also be rigorously validated to prove its ability to achieve sterility without negatively impacting the component's integrity or leaving harmful residues. The choice of sterilization method depends entirely on the material's compatibility and must be justified to regulatory authorities. Once sterilized, maintaining the sterility of the equipment and components until the point of use is critical. Sterilized parts are often wrapped in special packaging or stored within pre-sterilized containers. They must be transferred into the Grade A aseptic core using a validated method that does not introduce contamination. This is often done through a double-door autoclave that opens directly into the aseptic area or via a sterile transfer port. Every step, from initial cleaning and wrapping to sterilization and transfer, is governed by detailed standard operating procedures, or SOPs, to ensure that the machinery of sterility functions flawlessly and delivers a sterile pathway for the drug product. The aseptic process begins long before any filling operation. The first step involves the preparation of the sterile drug product itself. This bulk solution is typically formulated in a controlled environment, like a grade C area, and then passed through a sterilizing grade filter into a sterile holding tank. This filtration step is, honestly, critical. The filter has a pore size, typically 0.22 microns or smaller, that is small enough to physically remove bacteria from the solution. The integrity of this filter must be confirmed both before and after use through a validated test, such as a bubble point or pressure hold test, to ensure it was not compromised during the process. Simultaneously, the primary packaging components, the vials, syringes, or bottles, along with their corresponding stoppers and seals, are prepared. Glass vials are typically washed with highly purified water and then depyrogenated and sterilized in a dry heat tunnel. This high-temperature process not only kills all microorganisms but also destroys pyrogens, those bacterial endotoxins that can cause a fever response in patients. The tunnel is designed to directly connect to the filling line within the Grade A zone, creating a seamless, enclosed path that prevents the recontamination of the sterile vials before they are filled with the drug product. The core of the process is the filling and stoppering operation, which must occur entirely within the protected Grade A environment. The sterile drug solution is pumped from the holding tank through sterile tubing to the filling needles, which dispense a precise, predetermined volume into each container. Immediately after filling, a sterile stopper is mechanically placed into the neck of the container. This entire sequence is highly automated and occurs under a continuous stream of HEPA-filtered air. Operators in the surrounding Grade B area monitor the process, but their direct interaction with the open containers is minimized or eliminated through the use of barrier technologies like RABs or isolators. The final step is capping or sealing the containers to ensure their long-term integrity. After the stopper is inserted, the vials move out of the most critical aseptic zone to a capping station. Here, an aluminum overseal is crimped over the stopper and vial neck. While this step is considered less critical because the container is already sealed by the stopper, it is still performed in a highly controlled Grade A or B environment to prevent any surface contamination of the vial stopper interface. Once capped, the vials exit the clean room for inspection, labeling, and packaging. The entire journey, from bulk solution to sealed vial, is a carefully orchestrated sequence designed to exclude microbial contamination at every turn. The primary risk in any aseptic process is microbial contamination, which can originate from several key sources. As previously mentioned, personnel are the most significant threat. Even with full gowning, operators can release microorganisms through movement, speech, or an unnoticed tear in a glove or suit. Interventions, where an operator must reach into the Grade A zone to clear a jam or make an adjustment, are particularly high-risk events. Each intervention represents a potential breach of the sterile barrier. Therefore, a core principle of risk mitigation is to design processes and equipment that are so robust and reliable that the need for human intervention is eliminated or drastically reduced. The second major source of risk is the manufacturing environment itself. Inadequate air filtration, a failure in the HVAC system, or a loss of positive pressure can allow contaminated air from surrounding areas to enter the critical zone. Surfaces that are not properly cleaned and disinfected can harbor and grow microorganisms, which can then be transferred to sterile components. To mitigate this, manufacturers must implement a comprehensive and scientifically sound environmental monitoring, or EM, program. This program involves routinely sampling the air and surfaces throughout the cleanroom suite to detect the presence of microorganisms, providing an ongoing measure of the state of control of the facility. Contamination can also be introduced via the raw materials and components. If the drug solution is not properly filtered, or if the filter itself is flawed, the product will not be sterile. Similarly, if the sterilization process for containers or closures is ineffective, they will introduce contamination directly into the final product. Risk mitigation here relies on rigorous validation and control. Sterilization cycles must be validated, filter integrity must be tested for every batch, and suppliers of raw materials and components must be qualified to ensure they meet stringent quality standards. Every material entering the sterile path must have a certificate of sterility or be subjected to a validated in-house sterilization process. To manage these risks systematically, the US FDA encourages the use of formal risk assessment tools, such as failure mode and effects analysis, or FMEA. This involves breaking down the entire aseptic process into individual steps and asking what could go wrong at each point, what the consequences would be, and how likely it is to occur. By identifying the highest risk steps, manufacturers can focus their resources on implementing the most effective controls. For example, a risk assessment might identify a manual connection of two sterile pipes as a high-risk activity, leading the company to redesign the system with a pre-assembled, single-use, sterile welded tubing set to eliminate that risk. Given the profound impact of human behavior on product sterility, the training and qualification of personnel are among the most critical components of a successful aseptic processing program. The US FDA guidelines state that all individuals involved in sterile manufacturing, including operators, maintenance staff, and quality supervisors, must receive training specific to their duties. This training goes far beyond simply reading standard operating procedures. It must include a foundational understanding of microbiology, contamination control principles, and the potential impact of their actions on patient safety. They need to know not just what to do, but why they are doing it. The initial training program for a new operator is intensive. It covers basic cleanroom theory, hygiene practices, the proper way to clean and disinfect surfaces, and most importantly, the complex techniques of aseptic gowning. After classroom instruction, trainees undergo hands-on practice, often in a simulated environment, until they can demonstrate proficiency. Aseptic gowning qualification is a formal process where trainees must successfully gown and then have their gloved fingertips and suit sampled for microorganisms. They must pass with results below a predefined limit before they are ever allowed to enter the actual aseptic manufacturing suite. A cornerstone of operator qualification is the process simulation, commonly known as a media fill or aseptic process simulation, APS. In a media fill, the actual drug product is replaced with a sterile microbiological growth medium, like soybean casein digest medium. The operators then run the entire aseptic process from start to finish, including all assembly, filling, and manual interventions that would occur during a normal production run. The filled units are then incubated for at least 14 days. If any unit shows microbial growth, it indicates a breach in aseptic technique. A successful media fill with 0 contaminated units provides the highest level of assurance that the process and the operators can produce a sterile product. Operators must participate in these media fills, typically twice a year, to maintain their qualification. Training is not a one-time event, but an ongoing process. Regular refresher courses are required to reinforce key concepts and communicate any changes in procedures. Any operator who is observed performing a task incorrectly or is associated with an adverse environmental monitoring trend must be retrained and requalified. The training program must also cover how to respond to unusual events, such as a spill, a power failure, or a breach in the clean room barrier. This continuous cycle of training, qualification, and performance monitoring forges a chain of competence, ensuring that every person entering the sanctum of aseptic processing is fully equipped to protect the product's sterility. In summary, mastering aseptic processing is a fundamental requirement for the modern pharmaceutical industry. It is a discipline built upon an unwavering commitment to preventing microbial contamination in sterile drug products that cannot be terminally sterilized. The core principles, as outlined by the US FDA, are not merely suggestions, but strict requirements for ensuring patient safety. These rules revolve around the comprehensive control of every variable that could possibly introduce a microorganism into the product. This begins with the very design of the facility, which must use classified cleanrooms, unidirectional airflow, and pressure differentials to create a fortress against contamination. The human element remains the most critical and challenging aspect to control. The success of the entire operation hinges on the discipline and training of the personnel who work within this controlled state. Strict, validated gowning procedures are essential to contain the particles and microbes generated by the human body. Once inside the aseptic area, operators must adhere to a code of slow, deliberate conduct, always respecting the first air from the HEPA filters and minimizing any interventions that could compromise the sterile field. This human factor is so important that operators must prove their competence through rigorous qualification programs, including periodic participation in media, fill simulations. Equally important is the proven sterility of all equipment and components that come into contact with the drug. Validated sterilization processes, whether using steam, dry heat, or other methods, provide the necessary assurance that these items are free from microbial life before they are introduced into the aseptic core. This, combined with a robust environmental monitoring program that continuously verifies the state of control of the air and surfaces, creates multiple layers of protection. Risk management principles are woven throughout, pushing manufacturers to proactively identify potential failure modes and design them out of the process. Ultimately, the promise of sterility is upheld not by a single action, but by the flawless integration of a facility, validated processes, and highly trained personnel. It is a system of overlapping and redundant controls, all working in concert to achieve one goal: to ensure that every single vial of medicine is and remains sterile. Adherence to these fundamental rules, controlling the environment, managing personnel, sterilizing components, and validating the process, is the only way to safeguard the health of patients who rely on these essential, life-saving therapies. The meticulous world of aseptic processing is, in its essence, a profound expression of the industry's duty of care.